Hello all. In this video we are going to see about photorespiration. It comes under unit 6 of plant physiology and you are watching this on Life Science Library. First we will see what is photorespiration. Photorespiration is nothing but a process in which that uptake oxygen and release carbon dioxide. It is a unique process that occurs in plants, mostly in C3 plants. It is not seen in C4 plants because C4 plants undergo carbon dioxide enrichment pathway. So it is seen in C3 plant and not in C4 plant. Major enzyme involved is Rubisco. This is an important previous year question in CSIR that it is seen in C3 and not in C4. It is called as Warburg effect. This is also previous year question. What is Warburg effect? Warburg effect is nothing but carbon dioxide that is used for photorespiration will go and inhibit photosynthesis. So carbon dioxide consumed during the process of photorespiration will go and inhibit photosynthesis. So photosynthesis is deteriorated. Rubisco is an enzyme that has two different activity. One is carboxylase activity seen in C3 cycle, that is Calvin cycle, and the other is oxygenase activity, that is in photorespiration. That is oxygenase activity. In photorespiration, we will see about the oxygenase activity of Rubisco. This process of photorespiration is activated only if oxygen content is high and carbon dioxide content is low. What are the three major organelles that are involved in photorespiration? It includes chloroplast. Sorry, it is not peroxide. Very sorry, we apologize for that. It is peroxisome. It is peroxisome. Peroxisome, mitochondria. These are the three organelles that are involved in photorespiration: chloroplast, peroxisome and mitochondria in order. CPM, chloroplast, peroxisome and mitochondria. This is a double-walled membrane. This is a single-walled membrane, single bound organelle, and it is a double-bound organelle. That is, mitochondria is a double-bound organelle. Chloroplast, peroxisome and mitochondria. Now we will see about the entire cycle of photorespiration. Before that, we will see what will happen if oxygen is taken in and what are the major function of this Rubisco enzyme. RUBP is oxygenated. Rubisco enzyme is involved. It produces two different components. That is, one component is 3-phosphoglycerate. 3-phospho. If RUBP is oxygenated during photorespiration, it forms two different component, that is 3-phosphoglycerate and 2-phosphoglycolate Hope clear, my handwriting is clear. Very sorry for bad handwriting. So RUBP is oxygenated. It forms two different components, that is 3-phosphoglycerate and 2-phosphoglycolate. This 3-phosphoglycerate will enter dark reaction, that is C3 cycle, Calvin cycle, and 2-phosphoglycolate will undergo glycolate cycle, that is photorespiration. C3, it will undergo C3 cycle, it will undergo photorespiration. So 2-phosphoglycolate is the major component that begins photorespiration. Enzyme involved in oxygenation is Rubisco. Under oxygenase activity, 3-phosphoglycerate and 3-phosphoglycolate. Glycerate will undergo C3. Glycolate will undergo photorespiration. It is also called as C2 cycle. So now we will see about C2 cycle, that is photorespiration. What will happen in C2 cycle? So in C2 cycle we have phosphoglycerate and RUBP. We have seen now that when RUBP is oxygenated, it forms two different components, that is 3-phosphoglycerate will undergo Calvin cycle and 2-phosphoglycolate will involve in C2 cycle. 2-phosphoglycolate, a pi compound is removed, it forms glycolate. Glycolate from chloroplast enter peroxisome. Peroxisome, the glycolate is converted into glyoxylate. Here hydrogen peroxide is formed in this step. Glyoxylate is converted into glycine. Glycine is an amino acid. It is formed in peroxisome. Glycine enters mitochondria, where glycine is converted into serine. Serine is also an amino acid. Glycine is converted to serine, where ammonia is evolved, carbon dioxide is evolved, NAD+ is converted into NADH2. Serine enters peroxisome. Serine is converted into N-hydroxypyruvate. Pyruvate is again converted into glycerate. Here NADH is degraded to NAD+. So what is the reaction here? The reaction here is reduced. NADH is reduced to NADH2. It is actually NADH2. NADH2 is reduced to NAD+. And it forms glycerate. Glycerate is converted into 3-phosphoglycerate. Here ATP is consumed. ATP is consumed. So energy is not formed in the cycle, energy is consumed in this cycle. So the cycle is not an energy producing cycle. This cycle is actually a wasteful process. So the energy is spent releasing carbon dioxide and it is not releasing oxygen. This is the major gist. And here NAD+ forms NADH2. Here NADH2 is reduced to NAD+. This will be asked in CSIR exam. This will be asked in CSIR exam where hydrogen peroxide is produced, that is glycolate to glyoxylate. This is a previous year question. Then glycine forms serine. It occurs in mitochondria. This is a previous year question. So please make a note of it. Hope this C2 cycle is clear. As I said, this is a wasteful process. As I said, it occurs only in C3 cycle and not in C3 plants and not in C4 plants, because the C4 plants will have another strategy called as, we will see that when we see about a C4 cycle, that is the carbon dioxide enrichment pathway. In this pathway, they will make carbon dioxide content high inside the plants. So oxygenase activity of Rubisco is switched off. Carboxylase activity will begin. So it is not occur in C4 plants, it occur only in C3 plants. Temperature factor. High temperature promote oxygenation of Rubisco enzyme. Because of this solubility factor, of carbon dioxide, solubility of carbon dioxide declined due to high temperature. So oxygenation is promoted. Solubility of carbon dioxide is declined. Temperature increases, oxygenation increases. Why? Because solubility of carbon dioxide is decreased. So oxygenation is promoted. Next, Rubisco will be more active in oxygenation. Second, specificity of factor of Rubisco also decreases with increase in temperature. That is inversely proportional. Increase in temperature, oxygenation activity of Rubisco. Increase in temperature decreases Rubisco. Increase in temperature, promotion of oxygenation, hence the photorespiration, but Rubisco act inversely with increase in temperature. Increase in temperature has negative effect of oxygenation of Rubisco. These are the basic details and the clear-cut explanation about photorespiration. Now we have seen what is photorespiration, what is Warburg effect, previous year question, oxygenase activity of Rubisco, previous year question, organelles involved, previous year question, and what are the components formed in each organelle, previous year question, CSIR in case of CSIR exam. Then we have seen all the organelles and all the components formed in each organelle. Then about the factor of temperature. Increase in temperature increases oxygenation, whereas increase in temperature has inverse effect on oxygenation of Rubisco enzyme. Hope this video is clear and hope this video is useful to everyone. If you like this video, please do like, share and subscribe to my channel. Thank you for watching this video. Thank you.